Okay, so now what we're going to learn today, we're going to look at this diabetic mellitus. We have type 1 and type 2 diabetic mellitus, diabetes mellitus, sorry. So what are the differences between type 1 and type 2, and what how we can manage this type 1 and type 2, and how we monitor the type 1 and type 2 diabetic mellitus. Okay. So diabetic mellitus is a group of metabolic disorder. We call it metabolic disorder, characterized by this, what we call the high blood sugar, and this sugar actually referred to glucose level over a prolonged period. Okay. So what are the symptoms? Symptom, particularly you can see that frequent urination, increase the thirst, and increase appetite. Frequent urinations generally happen at night. Okay, you can see that when you go to toilet a few times one night. Okay, that's a symptom of the diabetes mellitus. Are you clear? So perhaps you need, a person may need to go and check whether he or she has the diabetes mellitus or not. So there are three main types of diabetes mellitus, but for us, we only need to know type 1 and type 2. Type 1 previously referred to the insulin-dependent diabetic mellitus or juvenile diabetes. So type 1 diabetes, basically, the onset is relatively earlier. It's a genetic defect sometime. Okay, so type 1 actually also called insulin dependent because type 1 means that they do not produce, the patients for type 1 diabetes, they do not produce sufficient insulin. So therefore, the blood glucose level is high until the glucose appears in the urine. Are you clear? Okay, type 2 diabetes. Type 2 diabetes actually refer to a non-insulin dependent diabetes mellitus last time previously, or we call adult onset diabetes. Adult onset, why we call adult onset? Because generally type 2 diabetes actually will be slightly later, the onset. It's not immediately. You can see the immediate, okay? But now diabetes, because of the diet, because of the lifestyle, you can see that even though type 2 diabetes, the onset now pretty earlier, some kids actually develop also this type 2 diabetes. So what's type 2 diabetes? Type 2 diabetes is more to the diet, more to the lifestyle. Are you clear? Okay. And body does produce sufficient insulin. Your body, your pancreas produce sufficient insulin, but the insulin receptor actually insensitive or resistant to the insulin. It means that insulin is there, but the insulin may not be able to bind, or the insulin receptor actually cannot respond to the insulin. So what is the difference of, what are the differences between type 1 and type 2 diabetic mellitus? So basically, the characteristic, type 1, we say that is insulin deficiency. Not enough insulin. Type 2 is insulin resistant. Means that insulin is there, but okay, the body actually pull in the response. Okay, how about onset? When is the onset? Diabetic type 1 usually very early in life. Okay, for type 1. Type 2 actually relatively late in life. Now, even though I say relatively late in life, doesn't mean that it only occur when you are age of 30, 40, 50, because it depends on the lifestyle. Okay, so what is the underlying factor? Okay, a person have diabetic mellitus type 1, normally is due to the autoimmune disorder. So what's autoimmune disorder? Autoimmune disorder basically means that our own immune system actually attack our own cells. Are you clear? Okay, so how they attack? They attack beta cells. Because we do know that beta cells produces insulin, right? Beta cell produces insulin. So if the insulin producing beta cell are attacked by the immune system, then therefore the cells actually have lost the capability to produce insulin. Can I see that? Okay, and also may due to the lack of an insulin coding gene. So it means that the gene is not there to code for the insulin, or mutation take place onto the insulin gene. Okay, so what are the treatments? No choice, because the lack of insulin, we provide additional insulin. So insulin injections is one of the way. Okay, type 2 diabetes. Type 2 diabetes, as I said, that they're insulin resistant. So due to what? Due to obesity. Okay, so due to the change in the insulin receptor, it might change, it might be covered by the lipids. Okay, the receptor actually lacks of the responsiveness in the target cell. Are you clear? So perhaps, for example, covered by the lipids, there's a change in the shape because of the obesity. Therefore, you can see that for type 2 diabetes, we don't provide additional insulin, no point, because body, the beta cells do produce sufficient insulin. So one of the ways is to exercise and dietary control. Exercise means that we try to absorb more glucose to the muscle cells, to the cells that we need. So when you exercise, you use up the glucose. Or dietary control, basically you get less glucose in your diet. Okay, so there's a difference between type 1 and type 2 diabetes. Only type 1 diabetes patients need to have the insulin injection. Now, just now we talk about what the type 1 and type 2, but I left out this gestational diabetes. Gestational diabetes basically is occur, this diabetes occur when the pregnant woman without a previous history of diabetes develop a high blood sugar level. So during pregnancy, don't know how, don't know why, this girl or this woman suddenly gets this diabetes. And this diabetes we call gestational diabetes. Need to be treated, need to be monitored. If no, after delivery, these high blood sugar levels still may remain inside this woman. Are you clear? Okay. So this is what actually happens. In diabetic persons, uptake of glucose into the cell is very slow because they can't respond to insulin, or insulin is not there, even though they have the plenty of glucose in the blood. So the cell actually now lack of glucose, particularly liver cell, start to metabolize the fat and the protein as alternative energy source. Are you clear? Our main energy source is the glucose, but now lack of glucose, the cells can't take up a lot of glucose. What will happen? That happens here is the cells start to use the fat and the protein as an alternative energy source, and it will leads to the build-up of the keto acid or ketone. This is acidic. Okay, this is acidic. So it will decrease the blood pH. So the combination of dehydration, salt loss, and the low body blood pH, it can lead to coma in the extreme situation. Okay, if not, it's not treated. But a person with untreated diabetes also may decrease steeply when the blood glucose concentration between the meal. Now, why? Because during the high carbohydrate meal, they can't absorb glucose, they can't store glucose. So means that they don't have the glycogen to mobilize when they lack of the glucose. Okay, so coma may result because lack of the glucose for respiration. Okay. So you look at these diagrams, okay? So we do have one of the term called renal threshold. I'll explain this renal threshold when it comes to the kidney, okay? So kidney have the ability to reabsorb back the glucose, but there must be lower, okay, or at particular, or is it that? Lower than a particular concentration. This concentration actually is 180. Okay, this concentration is 180. So if the blood glucose concentration is higher than 180, then you can see that glucose will be released in the urine. So if you look at these diagrams, initially our blood glucose level is low. So after the glucose injections, that is increased, but no matter how it increased, we won't be over 180. And then because of insulin, can I see that insulin, non-diabetic patients, so therefore glucose concentration drop, and then we monitor them back. Okay, but for those diabetic patients, you can see the glucose concentration shoot up high and above 180. When you're above 180, what will happen? If above 180, means that some glucose won't be able to be reabsorbed back into the blood. So therefore, glucose appear in the urine. Then we can actually check the presence of the glucose in the urine. Are you clear? So that is one of the indicators whether a person developed type 1 diabetes or not. okay, or even type 2. Okay, so we check this. So this one, because that we can see that the diabetic patient here, blood insulin level is low. So this person must be type 1 diabetes. If type 2, you can see that they produce sufficient insulin as well. So whether non-diabetic or diabetic patient, insulin level is the same. So if we check the insulin level, okay, normal, then this person has type 2 because urine got the glucose. Okay. So how we manage the diabetic mellitus, okay, how we manage the diabetes mellitus. So the management of diabetes mellitus revolve around keeping the blood glucose concentration constant. So how we can do this? We can actually monitor the blood glucose concentration, okay, using the urine glucose dipstick. Now this is not so true because you can't monitor blood glucose by using urine glucose dipstick. But by using urine glucose dipstick, we can some sort of the indicator to check on the blood glucose concentration, okay? Or we can use a glucose biosensor. And for type 1 diabetes patient, we can use the insulin injections to make sure that they have the low or normal blood glucose concentration. Now we look at the first one, a urine glucose dipstick. So the enzyme here, glucose oxidase. So if you look at this urine dipstick, you can see that we have this, what we call the test region here. So on this pad, okay, if you look at it from the side, they do have the immobilized enzyme. So this immobilized enzyme is known as glucose oxidase. Are you clear? Okay, glucose oxidase. So what's this immobilized enzyme do? This immobilized enzyme can convert the glucose together with the water in the urine so that to give us a compound called glucanolactone with the side product produced here, hydrogen peroxide. But you look at this, glucose, water, oxygen, glucanolactones, and also hydrogen peroxide, all of them, they are colorless. Okay, they are colorless. So how we can make it color? In this case, in this pad, we also add in the colorless material called potassium iodide, Ki. So we also have Ki inside with another enzyme called peroxidase. So the second enzyme called peroxidase. So with the peroxidase and K1, what will happen here is peroxidase catalyze the conversions of the hydrogen peroxide with a reaction between the hydrogen peroxide and the Ki or potassium iodide to get a brown compound. Are you clear? Get a brown compound KiO3. So in our syllabus here, you just need to know with the presence of the hydrogen peroxides, peroxidase will be able to catalyze the reaction between the hydrogen peroxide and a colorless compound to get a brown color compound. So the intensity of this brown color compound will be indications for us to know how much glucose we have in our urine. So the darker the brown color means that the higher the concentration. So what we can do, actually, we can match the color with the reference here. So for example, if the color a little bit like this, okay, it'll be green brown in this case, then it's about 2%, okay, about 2% of the glucose. If let's say get about like red brown in this case, okay, then we do know that that is the highest concentration. So it's something like our semi-quantitative, okay, semi-quantitative, okay, Benedict test, okay, semi-quantitative Benedict test, where we actually get a range of the glucose in our urine. Okay, so the darker the color means that's the higher the concentration. Okay, so I put against the enzyme that we need to remember here, glucose oxidase. Okay, glucose oxidase is the one that converts this glucose, okay, into glucanolactone. And glucanolactone actually is an acid. Okay, glucanolactone means acidic here. Okay, so some books actually use the gluconic acid to replace the term called glucanolactone. Okay, okay, the gluconic acid. Okay, either you say glucanolactone or gluconic acid. Okay, now, next, we look at the biosensor. Biosensor is the device, something like this. The device, something like this. So we have something like a chip. So we prick our finger, and then we get a drop of blood, and then we slot it into this machine. We will be able to read the concentrations of the glucose. Okay, so biosensor again is an electronics monitoring device. Again, use the biological material, for example, a cells, enzyme, or antibody to detect or measure the chemical compound. And why we use enzyme antibody? Because it's specific and can detect a particular molecule in the complex mixture. Okay, so what's the principle? You look at the principle, it's very, very simple. So we do have the immobilized biomolecule here. It can be enzyme. So in glucose biosensor, we use the enzyme, same enzyme, glucose oxidase. Okay, glucose oxidase. Okay, so you can see that we can actually have our analytes, means the blood. The blood contains many, many different compounds, right? But glucose can bind to this enzyme. So when the glucose binds to this enzyme, okay, and then it will produce this glucanolactone. As I said, that glucanolactone is acidic. So it means that, can you see that we change the microenvironment in the biosensor, and this bioreceptor will be able to pick up this, what we call this, the change in the microenvironment. Therefore, we can use a transducer to convert whatever signal that we have into the electrical signal, and we amplify. We will give us the data processing and allow us to read or get the value, right? The exact value of the concentration. Okay, so how we can do this? Because we do know that if the glucose is converted to glucanolactones, they're acidic, so mean that there is a release of the H+ ion. So this H+ ion will give us a different in terms of electrochemical. Initially, no H+ ion, but now with the present H+ ion, so with the H+ ions, it will cause the change in the electrochemical. So this electrochemical change will be picked up by the transducer, and the transducer will convert it into the electricity, okay, microelectricity, and amplify. Then it will give us the reading. So it means that the highest the concentration of the H+ indicates that there is higher concentration of glucanolactone. So therefore means that glucose is in higher concentration. So it totally depends on how much glucose available in the sample. And the current is detected by the electrodes, amplified, right? I say that, by the meter, which produce a reading for the glucose concentration. So the more glucose present, the greater the currents, therefore the greater the readings from the biosensor. Okay, so glucose attached, against glucose attached to this or bind to the active sites of the glucose oxidase, and the glucose oxidase now can convert the glucose into glucanolactone. So this glucanolactone is acidic, so therefore they actually release the H+. So this H+ actually cause the change in the electrochemical electrochemistry here. So therefore the transducer will be able to convert this change into the electricity. But this electricity actually is very, very minor only, or a minute intensity. We need to have the amplifier to amplify this intensity so that we will be able to read or to give us the exact reading. Okay. Now, what are the pros and cons by using this urine glucose dipstick compared to glucose biosensor? Okay. Now, if you look at advantages here, urine glucose dipstick, the collection of urine is a non-invasive method. You do not need to prick. Can I see that? Are you clear? We do not need to prick our body to get the blood sample. We just urinate and then you get. So when I say non-invasive, normally we follow by the idea here is decrease the risk of infection. You just pee, right? You just urinate. So it means that you don't need to worry about the bacteria or any viruses enter into the wound because there's no wound there. Okay? And if you compare to the Benedict test, You compare to the Benedict test, therefore, it's quite fast. If within 30 to 60 seconds, you get the result, right? If Benedict test, you need to heat up, okay? And the strip are relatively cheaper as compared to the biosensor. They will later look at this, okay? But what is the disadvantages? The test is a semi-quantitative, means it gives us a range. It won't give us an exact value, okay? The reading of the color is subjective in this case, right? You read the color, the way I read the color may be slightly different, and the color may be in between the range. So therefore, we won't be able to actually judge. Let's say, for example, between 2 and 5%, then we won't be able to actually give a very exact value. And the test does not indicate the current blood glucose concentration because it's urine. Urine means that you need to pass the renal threshold before you can see the glucose spilled into the urine. But it doesn't mean that if, let's say, you check using the strip, oh, there's no glucose in the urine, but it doesn't mean that your body actually maintain a normal range, okay? Because normal range should be 80 to 120. So if higher than 120, 150. If 150 means that haven't reached the renal threshold, but your blood glucose level is high. Can you see that? So it won't give us the current blood glucose concentration. That is a disadvantages. Okay, one of the disadvantages. How about glucose biosensor? It's a rapid in process compared to Benedict tests, okay? And it gives us a specific digital reading instead of a range. Okay, a specific digital reading, 100, 89. Okay, so this is what means that by give us the digital reading rather than a range. And the test does not need to interpret the color chart. So it means that you don't have the problem of arguing, oh, I think this is a dark brown, or this is a red. You don't need to argue because it gives us straight away the digital reading. The test here indicates the current blood glucose concentration because we use a blood sample. So therefore, it will give us the current blood glucose concentration. Okay. And the device or the biosensor, not the strip, but the device can be reused, okay? But the dipstick cannot be reused, okay? Now, collection of blood sample require a finger prick. So therefore, there is a risk of the infections for disadvantages here. And biosensor, basically the device is more expensive compared to the dipsticks, okay? So highlight this. Normally, when a question asks about glucose dipstick and glucose biosensor, they will ask you to compare between these two, okay? Any more questions so far, guys? So we come to the last part here, okay? Before we go to adrenaline in the blood glucose regulations, I did include what the part here is the human, okay, insulin. Produced by the genetic engineering. Okay, this one I have done it before already, right? Okay, why we use a human insulin compared to the animal insulin? So we have done this in the gene technology. Are you clear? So one of the way, as I said, that not only monitor, but for the type 1 diabetes patients, we give them the genetically engineered human insulin. Correct? So the benefits, I won't going to talk about it here. For example, a rapid response, less allergic reactions, can have mass productions, and then less ethical issues and blah, blah, blah. So those you can refer back to the gene tech note. Okay, so I won't go into details now for the treatment.